Scene 1. Introduction prompt. Create a professional 3D medical animation in a modern laboratory. Show a DNA double helix rotating slowly. Hindi title appears. PCR polymerase chain reaction. Kya hai? Clean educational style. Realistic molecular visualization. Bright laboratory lighting. High quality scientific animation. Scene 2. Sample collection prompt. Show a scientist collecting a biological sample in a laboratory. Display a test tube containing DNA sample. Hindi text. Charan 1. DNA namuna ekatra karna. Professional educational animation. Detailed laboratory environment. Scene 3. PCR reaction mixture preparation prompt. Show a PCR tube being prepared. Add DNA template, primers, nucleotides, DNTPs, tuck DNA polymerase enzyme, and buffer solution into the tube. Labels appear in Hindi. DNA template, primers, tuck polymerase, DNTPs, buffer, scientific 3D animation. Scene 4. Denaturation 95 degrees Celsius. Prompt. Zoom into DNA molecules inside a thermal cycler. Temperature rises to 95 degrees Celsius. The DNA double helix separates into two single strands. Hindi text. Charan 2. Denaturación 95 grad. DNA की दोनों strength अलग होती है. Realistic molecular animation. Scene 5. Annealing 65 degrees Celsius. Prompt. Temperature decreases to 55 degrees Celsius. Primers move and attach to complementary DNA sequences on single-stranded DNA. Hindi text. Charan 3. Queen. Annealing 65 degrees Celsius. Primers DNA से जुड़ते हैं. Detailed molecular visualization.
Welcome to this educational breakdown of the final key stages of the polymerase chain reaction, or PCR. We begin at scene 6, held at a constant 72 degrees Celsius. Here, the Taq DNA polymerase enzyme gets to work. It moves along existing DNA strands, adding complementary matching nucleotides one by one. This builds new, full, complementary DNA strands, as shown clearly on your screen. Moving to scene 7, we see a professional infographic cycle diagram mapping the full PCR process. Denaturation, annealing, and extension repeat over and over. After each full cycle, the number of DNA copies doubles, growing exponentially. 2 become 4, then 8, 16, 32, and finally 64 copies in just a few rounds. Scene 8 fills the screen with a clear scientific visualization of millions of bright new DNA copies. This is the final result. After 25 to 35 cycles, PCR produces hundreds of thousands of identical target DNA copies. Scene 9 uses a modern split-screen animation to show 4 key vital real-world applications of PCR technology. These are disease diagnosis, forensic DNA testing, genetic research, and pathogen detection, each clearly labeled. This final screen summarizes the full PCR workflow. PCR is a fast, accurate method to copy DNA.